Hello fellow biologists, today we are looking at lesson objective I taken from the OCR specification for A-level biology, cloning and biotechnology. We're going to be looking at the uses of immobilised enzymes in biotechnology and the different methods of immobilisation. Got to look at the different methods involved and also an evaluation, so we're looking at advantages and disadvantages of using them. And there are some examples that could include, so when it says could, you don't really need to learn these off by heart. There are a couple of slides at the end that you can pause and read over to get an idea about how you can actually use immobilised enzymes in biotechnology. Okay, so immobilised enzymes, as it sounds, the enzyme are attached to an insoluble medium or material, and therefore at the end they're not mixed up in with the products. They're easily separated out. And there are some advantages and disadvantages of doing this, which we're going to move on to in the evaluation part. So the different methods, you do need to be aware of these. The bits in the red boxes are taken from the mark schemes. So the first one is encapsulation, where the enzyme is trapped within inside a semi-permeable membrane. So the substrate can still get in and bind to the active site, creating an enzyme substrate complex. The enzyme can still break it down into products, and the products are released. Entrapment, so this is where the enzyme's trapped inside some silica gel. Same thing, substrate can still get in, bind to the active site, so it's complementary and specific, form an enzyme substrate complex and release the products. We have cross-linked here because don't forget an enzyme is a protein and it has it's made of amino acids, so we can actually form covalent ionic bonds between the amino acids and the enzymes using this chemical here. Apologies about the typo. And the last one is the carrier bound, where the enzyme can be attached to a carrier such as activated carbon or clay. Again, keeps the enzyme in place so it can't be mixed in with the final product, because we want to separate the final product and the enzyme. It makes it better for downstream processing. So here are some advantages. These are taken from the mark schemes. So the enzyme can be easily removed to be used again. There's less downstream processing, as I mentioned before. It leads to a purer product because the enzyme doesn't need to be extracted from the product. And we also have that they're more stable, these enzymes, in immobilised enzymes, because they're less likely to be denatured by pH or high temperatures, because the immobilisation of them, it kind of protects them. So, for example, if they're protected by a membrane or clay or carbon products, they're just a bit more protected from the elements, so to speak. So disadvantages, these ones are not taken from the mark scheme. But they do make sense. So the process of immobilisation of an enzyme can reduce its activity, that can't mix so freely, higher initial cost, a higher cost of immobilising enzymes compared to free enzymes. There's also a more complex procedure involved. So we've looked at now the methods of immobilising enzymes, different methods. We've also looked at evaluation, so advantages and disadvantages of using them. And here are some examples that we need to be aware of. So I'm not going to read these out, but I'll flick through so you can pause the screen and have a read of the material, okay? Okay, so that's the information that you need to know on immobilising enzymes in biotechnology. Guys, good luck with your exams. Don't forget to do not use the words amount, it, they, size. And good luck with your exams and your revision.